Good morning, everyone. Welcome to the February 2025 Zoom Citrus Seminar. Thank you all for joining us. First, I would like to thank Alison Walton from the Mongi IFAS Center for her help and cooperation. Today's program offers one CEU for pesticide license renewal and one CEU for certified crop advisors. If you need CEUs, email me your name, email address, and license number. Our guest speaker is Dr. Jude Grosser, Professor in Citrus Breeding at the UF-IFAS Citrus Research and Education Center in Lake Alfred. The title of his presentation is Option for Planting Profitable Citrus Now and in the Near Future. The presentation will focus on new HLB-tolerant high-quality sweet oranges and juice blending candidates as needed to save our NFC juice industry. Focus will also cover the best new HLB-tolerant rootstocks to combine with the new scion. Information on other new citrus candidates, including red grapefruit, seedless lemons, and a hybrid lime, will also be presented. Finally, new high-value scions with potential for cups will also be discussed. Dr. Grosser, it is all yours. Please go ahead.
Here we go. Good morning, everyone. Of course, I'm representing our team, which includes Fred Gamitter, and I think he's listening in, and our newer members, Mongi, Jude, and John Chater, who's been making the rounds. And so you've probably encountered these two enthusiastic young guys that are now working with myself and Fred. So I don't have to tell you much about the crisis going on, but we're down to around 10% of normal production, and a lot of people are going out of business. So we need to be thinking about the fastest way to stem this tide right away. So first, I'm going to talk about true sweet oranges, as we've made a lot of progress on things that have a slightly or even a little bit significantly better tolerance than the traditional Hamlins and Valencias. So the quality of our NFC product is declining, the price is going up. So if we want to sustain a good market for our juice, we need to really address the quality of the juice. So I have a picture here of some OLL8 juice on the left, and the OLL series, of course, is named after Ori and Louise Lee, and with Ori Lee being the Hall of Fame grower that helped us develop the varieties. But then you can see some store-bought NFC on the right. We have an IFAS event at the Ben Hill Griffin Stadium every year called Flavors of Florida, and we always have a presence there. So the last 2 years, we've actually taken some juice and some fresh fruit samples there. But I have a photo to show you here. At the bottom of that picture is OLL8 juice that came from 4-year-old trees with HLB on UF-5 rootstock. And in the middle juice there is a 50-50 blend of Sugar Bell with the same OLL8. And then the poorly colored yellow juice there is from NFC that we bought at Publix. And so there's about 400 people that attend this event every year, and we have them vote on their favorite juices. And so last year, 5 out of 10 voted for the OLL8. 4 out of 10 voted for the blend of Sugar Bell with OLL8, and like 1 out of 10, only one person out of 10 prefers the NFC you can buy at Publix to juice from 4-year-old trees with HLB. So that should tell us something about how good the quality of the juice is. So the last OLL to be released before the two that I'm going to mention in a minute was OLL20. And it even has a more interesting flavor that was identified by Tropicana. So it was released because Tropicana asked us to release it. So here's some display data from the CREC fruit juice displays that we have 4 or 5 every year. And in this one, we were comparing the juice of 3 of the OLLs with a new clone of Valencia B965, and then some Sugar Bell blends. But what I want to point out to you is that the OLLs really do have good flavor, and Sugar Bell has a really good pasteurized juice flavor. But in this case, the OLL 20 is the winner for overall flavor. And we've never had OLL lose to Valencia in a taste panel, and 80 or 90% of the time they'll beat Valencia. So we found a couple of clones that are showing even better tolerance than OLL 8, and we just did a fast track release on those. They are OLL DC 336 and 340, and they were just released a couple months ago. Some fuzzy pictures here, but I've got my daughter standing next to some trees in a MAC trial at Eagle Lake, which is about 10 miles south of the CREC. These are 3.5-year-old trees on 942 rootstock planted at the same time. Everything's the same. You can see the difference, the fruit size, the crop, the health of the tree, the size of the tree. Everything you look at, the parameters are significantly different between these clones and the standard Valencia. And here's some data to back that up. There's statistical differences for just about every category of data you can look at between these OLLs and standard Valencia. So 2 of those are available, and they've been made available for craft, so you can get them planted out and get some help paying for the trees through the craft program. Here's a picture of the trees at Eagle Lake. This is from August of 2024. So you can see that they're uniformly nice to look at. They don't look like typical sweet orange trees of that age in Florida these days. So I want to talk about a new clone of Hamlin that was also just released. And you've already heard John Chater talk to you about N1332 because it does have enhanced HLB tolerance. This is another one that we've selected. Years ago, Fred and I evaluated over 1,000 clones of Hamlin, and we ended up selecting 3 clones. And this one was selected for high soluble solids. The reason that we didn't really push forward with these clones is because the EV early Valencias came along, and once we felt like we could have Valencia in the in the Hamlin harvest window, why would anybody want Hamlins? But now with HLB, the situation is a little bit different, and the EVs are struggling just like some of the original Hamlin clones that are commercial. So this one has kind of come back into play because it's still making higher bricks than the traditional Hamlin, and we know bricks. There's a bricks crisis in the entire industry. None of our trees are making the bricks that we've been accustomed to before HLB, so that makes it difficult to hit our bricks targets in our NFC juice, even with blending. So here's this Hamlin N1410. It actually comes in a few weeks earlier than standard Hamlin. It's a soma clone again. It's a regeneration from protoplast, so you can call it a protoclone, but it's derived from tissue culture. So there's some mutations that happen either through the tissue culture process or they're uncovered by the tissue culture process to give us this variant clone of Hamlin. These are some young trees at our trailer park block that have had HLB for several years, but they're generating fruit with higher bricks than the standard and higher bricks than the N1332 as well. So I pulled up some really old data and showed data from 4 different seasons here where we looked at a large number of Hamlin clones through our pilot plant data. You can see that the ranking for bricks production was always amongst the top, and 2 out of the 4 years it was the top for high bricks out of the clones that we evaluated. So it's always been one of the top out of the 1,000 for bricks and soluble solids, and that's carried through to the trees that have HLB as well. So here's some data from the trailer park trees, comparing it to the N1332, and you can see that the bricks is quite different, and the ratio is higher, which means you could pick the fruit a little bit earlier, and soluble solids is going to make you a lot more money at 5.56 versus 4.6 there for the 1332. So hopefully the tolerance will be just as good as 1332, but we got to get more trees out to determine whether that's true or not. So this has also been made available for craft. So if you want to try to get some trees through that program, go for it. Orange-like hybrids are of interest. There's a renewed interest in in-store juicing, so you want to have something that's attractive. Fred's got a number of really beautiful hybrids, and I've got a few too, but I'm going to show you our favorite one, which is this 1859. You can see that's what the color looks like. Doesn't need cold nights to make this color. The juice flavor is phenomenal. And so it's a candidate for fresh market, for in-store juicing, and for blending to make NFC juice better. So it's a hybrid of Sugar Bell LB89 crossed with a somatic hybrid of Nova plus Zekri. That the pollen parent was made in my lab. So Fred made this triploid, selected it in 2018. Here's one of the trees, the original tree, I think, in the field, showing some pretty good tolerance in 2021. Now you can look for several more of these kinds of hybrids coming along. Fred's working on the release of 104, and I've got one that's 18A435 coming along. And so these are going to be big opportunities. We just had the 104 juice in our last display, and everybody just raved about it. So I can't wait till we get the numbers back on how it compared to the other juices that we had out. We had some nice juices out to compare it to. So we've also been working on finding blending hybrids, because you're allowed to blend 10% mandarin juice into NFC. And so if you want to correct a bricks or a color problem, if you can find HLB-tolerant mandarin hybrids that have high bricks, high color, that can be blended, you can really fix a bad batch of juice just with 10%. There's an effort underway in the industry to get this increased to 15%, which would be good, because that increases your opportunities. We've made a lot of blends and put them out in our fruit displays, and a blend has pretty much never lost to a straight up juice. So we're missing the boat by not taking a stronger look at this. There's a lot of opportunity. So the work we've been doing with the Coca-Cola company, very extensive work. They've gotten heavily involved, and it's been a real godsend. But we've already started making selections based on their characterization of juice that's been pasteurized. They like the quality, what it can do to contribute to the quality of the NFC in a blend. And so we've got two selections here that I'm going to show you. One of them is a mandarin, triploid mandarin called C4 1042, which is another hybrid of Sugar Bell. Here's what it looks like. The downsides to it is the fruit is small, and it might be a little bit on the soft side, but I think it's tough enough to be trailered, but that remains to be seen. But this thing can make up to 18 bricks, even in trees from HLB. The bricks is usually coming in at 14 to 15, but it'll go up to 18. And you can see the colors off the scale. You can get up to a 42 juice color. So it doesn't take a lot of this to fix a batch of NFC juice that's slightly below what the target numbers are. This is another one that Weston Johnson at Coca-Cola really, really likes the juice attributes of. And it's a diploid hybrid that Fred made. The juice is just fantastic. This fruit's from a top work tree in a big trial that we have with them. And here's one of the top work trees, what it looks like, grown off in a very tough situation down in Zolfo Springs. And so here it is. This one's not quite through the parent tree program yet, but it will be very soon. It's a hybrid of Sugar Bell with Orkney. The fruit is larger and firm, so it's made for the truck. It can really do a lot for a batch of juice as well, and the bricks can get up to close to 14 with a nice color score. Another key factor of all these is having low limonene. Limonene seems to be a chemical that causes pasteurized juice to become bitter, and so it's an indicator whether you have bitterness in the juice. So we're finding a lot of really nice selections that are tolerant, but they have higher limonene levels, and these two that I'm showing you have lower levels that the juice companies are finding acceptable, which is an important thing. So now I'm going to talk about some mandarin hybrids, and for sure all of these work very well in cups. We're still trying to figure out how to grow them outside, and some of them are showing promise for outside also. So the first one is Marathon. Is a hybrid that Fred made. He named it Marathon because it has a very long harvest window, and it has super HLB tolerance. And the one thing I really like about it is it's different from a lot of things where you see beautiful trees, because you almost never see any symptomatic fruit on the trees. The trees are vigorous, they're productive with HLB, and the fruit's delicious. And it does have a sensitivity to alternaria, so you got to take care of that. But it's doing very well outside, and we've test marketed a little bit of fruit, and it's done very well. So if you have questions on this, you can contact Fred. Or if you want to see some mature trees, you can come on up and visit with us, and we've got a couple of rows of really beautiful trees that we can show you. It's a hybrid of daisy mandarin, and it gets its seedlessness from this Bukaku Kishu parentage. Here's cropping on the tree. The external color is not as dark as some of the other mandarin hybrids, but it does color up a little bit better when you treat it with ethylene. Next one is another one of Fred's 1956, RES 1956. This one's turned out to be really super interesting in cups. Forms fruit in these clusters. It's just beautiful. The fruit's on the small size. It would be on the small end of the cutie halo stream that you find in the store. But the fruit's super easy to peel and delicious. And the folks that have it in cups are pretty excited about it because it crops very, very heavy on small trees and can really produce a lot of fruit. Some information on it here from Fred, and it's again early maturing. It's something you can get in September. Does fantastic in our fruit displays. Everybody that tries it really likes it. Good candidate. Whether you can grow it outside or not, that still remains to be seen. But hopefully on the new rootstocks coming along now, we'll be able to do something outside with it. Here's a cups grown tree. Jerry Mixon that we're doing a lot of testing with. He's been a godsend as well. So again, you can see beautiful color without cold nights. So it's remarkable. Next one I want to talk about is triploid hybrid 18A939. We're calling it Gator Bites. It's a more recent release. In cups, this tree is fantastic. We top worked some trees in 2021. One of the trees made over 400 pounds of fruit this year, and the size distribution was really nice. It's got a cutie halo size, slightly bigger than the RES 1956 that you just saw. Very convenient to eat. We're getting rave reviews. People are telling us it's better than any cutie halo they ever have, and that's without soliciting the comments. In the cups, it's completely seedless, but outside with cross-pollination, when you got a lot of other citrus around, it will have 1 or 2 seeds per fruit. We have been able to get decent crops outside, but it requires really good nutrition. And I think once we get a little bit of improvement in the rootstock, we might be able to pull it off. This year, about 80% of the fruit outside was good to eat, and the animals are enjoying it right now, finishing off the fruit that was still remaining on the trees. In cups, it comes in December. Outside, it's been about a month later. This year was January maturity outside. Here's what it looks like outside when it's really doing well. This year the crop was lighter, but inside of cups, we're getting a crop like that just about every year. We've had it now going on 3 years. So a good one. Some other fresh fruit hybrids opportunities. I'm not going to show a lot of pictures of 914 because we've covered that in past presentations, but it's the dark red that doesn't interact with prescription drugs, and the flavor's off the scale good, and it's available. So let's see. This is a relatively new release called C253, and there's 2 other siblings coming right behind this thing, but it's really interesting. It's a triploid hybrid. you can harvest it in September. It'll have a 10 bricks and 15 ratio in September. So it's ahead of any grapefruit selection, true grapefruit selection that you can grow in Florida. It has better canker tolerance and better HLB tolerance than standard grapefruit. So I think it's something that more people could take a look at. The problem with it is we don't have a market for it, and there's not people packing grapefruit in September. So those kind of things have to be worked out. But I think if you get enough fruit, you'll find a market for it because people like it. We've released 4 seedless lemons, or what I call nearly seedless lemons. So you'll cut 10 fruits, you might find one or two that has a seed in it. They are all being made available, and I think they're going to help the dooryard market as well. But you know, lemons are more tolerant than most other scions to HLB, so there's an opportunity. And I think with seedless selections, there's a big advantage in the fresh market, because who wants your straw getting clogged with seeds? And if anybody does a lot of dishwashing with their dishwasher at home, and you drink a lot of water or iced tea with lemons in it, you always find seeds clogging up the jets in your dishwasher. So getting rid of the seeds is going to be a nice thing. So we've got 4 selections. One's from Eureka that we've named after a beautiful young lady that worked on this project for over 10 years, and we lost her sadly to ovarian cancer, but we named this first selection after her in remembrance. And we have a Todo del Año type, we have a Beers type, which of course is adapted to Florida, and we have a Lisbon type. These are all in the nursery right now for replicated trials, and we got trees that are going to be going in the ground this spring to take a larger look at these. We've also released a hybrid lime. It's actually a complicated hybrid that we're calling Fiestaville. It's very productive. We've taken this to the Flavors of Florida event and gotten super good feedback from the culinary experts there, and one chef in particular went crazy over it. So it's characteristically similar to a Mexican lime, but you don't find any dry granulated fruit. It's always very juicy. It's nearly seedless. You will find an average of about 1 seed. Again, when it's cross-pollinated, we don't know what it'll do when it's not cross-pollinated. And it comes off the tree already looking like it's been shined up. This fruit's straight off the tree without any kind of waxing. It's productive. So we're excited to have this, especially for the dooryard market as well. But the lime industry might also have an interest in it. We have a sibling of this that we're probably going to release as well, but it's not as easy to grow with HLB. But it has a very similar fruit, but there's a hint of mint in the flavor. So it has a very nice flavor too. We think these are going to have more cold hardiness because they have some kumquat in them, because limequat was one of the parents. So that's the characteristics that I've already pretty much told you about. One other thing I'll mention about it is it's kind of like a lemon, so it likes to flower all year round. So for a dooryard tree, it'd be really nice, because you're going to have fruit coming off multiple times during the year. So all those lemon clones are available, and you can order trees from your favorite nursery. All they have to do is get a license to grow the variety. So I don't know why I forgot to delete those. I want to mention Mongi Dut's got these 2 finger limes that are available, and you've probably heard him talk about them already. But they have beautiful colored caviar. They're both released. There's quite a bit of information on both of them. Of course, they're being used for garnishes and salads and to fancy up cocktails. Mongi has planted a rootstock trial, and the trees are starting to fruit, so he'll be able to give you some good advice on what rootstocks to use for these if you intend to grow any of these. So also available. All right, so I'm going to switch gears now and start talking about rootstocks, because I think the rootstock's going to be the answer to HLB. And keep in mind that if you you can find one or more rootstocks that can actually send something through the graft union to the top of the tree that can mitigate the disease, then you can grow whatever scion you want. You don't have to fix every scion. So this is the ticket. And I think we made a mistake at the beginning of the HLB crisis by not doing a Manhattan project to screen for rootstocks. So I've kind of, well, with the help of Fred and John and Mongi, we've kind of been trying to do that on our own here, and Ahmad Omar's been involved in this too. So we've been trying to screen thousands and thousands of rootstock hybrids, and I'm going to kind of show you some highlights of that. And then at the end, I'll just mention some data on some of the commercially available rootstocks, but the ones that I'm going to talk about in the next few minutes are just now going into large trials, and we still need to nail down which ones are good and get them more easily propagated for availability to the industry. So to have a rootstock that's better than something that's already commercialized, there's a whole long list of attributes that you need that I have here. And this list is growing. You need adaptation to all kinds of soils. There's tremendous soil variability in Florida. You know, there's still CTV, and now that we're backing off of psyllid control programs, you're starting to see more aphids. So CTV is still out there. It hasn't gone away. There's a lot of people planting sour orange. That's a risk that they're willing to take, that the CTV inoculum is down far enough and there's not enough aphids to spread it around. So they'll probably be able to get away with this for a while, but I don't know about the long term. We still have citrus blight. And of course, the Mandarin Pomelo hybrids have always done better than the trifoliate hybrids. The most important thing, high yields and good fruit quality. There's a lot of interest in mechanical harvesting. We just had the Agro-Malort demonstration day. The two rootstocks that were used in the trial were US-897 and UF-R6 that I'll mention. So optimum tree size is important, especially if labor becomes more of an issue. Diaprepes Phytophthora, that problem has not gone away and still causing a lot of issues for people. We also have sting nematode in a lot of groves in Central Florida. That's a huge problem. And now we have salinity problems in some areas. Some areas, the aquifers are getting salinized by ocean levels coming up. And we also have the problem of if you're irrigating out of a canal in a coastal area. I've had this actually happen to me. If you have a drought, the salinity in the canal can get up over 2,000 parts per million, and you're irrigating your trees with poison salt water. So this is going to be a growing issue. But number one, of course, formula bin. What can we do? So our program has always done conventional breeding at the diploid level, but we started making somatic hybrids by protoplast fusion. And so we did this to combine complementary rootstocks. And we made tetraploids. We started testing those in rootstock trials, and we found they had an interesting performance for various characteristics. And then some of these started to flower and fruit, and we learned that we could breed at the tetraploid level, which is very powerful because you can mix the genomes of 4 diploid rootstocks together at one time. So our toolbox for being able to do breeding of rootstocks has grown, and we're taking full advantage of that. So another thing that historically was necessary for a rootstock was for the rootstock seed trees to produce a lot of seeds, and also for them to produce a very unusual type of seed. Because when you think of seeds, you think, okay, it's a random half of mom's genes and a random half of dad's genes, and every seed makes a different baby. Well, for citrus rootstock propagation, the nurseries fell in love with rootstocks that produce seeds, that all the seeds produce embryos that are coming directly from maternal tissue, which are called nucellar embryos. And so they make clones of the mother. And so all the rootstocks are uniform, and you know exactly what you're going to get. So that's just a small fraction of the total genetics of the germplasm that's available. So you are restricting yourself to the amount of germplasm that you could use for rootstock improvement. So maybe the things that are tolerant or resistant to HLB don't produce that kind of seed, and they weren't getting utilized in the breeding program. But now that we have tissue culture micropropagation, we don't have to really worry about that anymore, because we've got several commercial companies, and we're working very closely with Agrimalora and Philip Ruck's nursery, who has the Genius Beth lamb working. And if you give them a new rootstock candidate, even before you have a seed tree, whether you even know whether it's going to produce seeds and whether the seeds are going to be the right kind or not, you can go ahead and get started on large scale micropropagation of that rootstock. Now, not all of them take off in tissue culture. And so if you put in 10, you might have 5 of them take off right away with their standard media, but some of them may require more work. And that's something that Beth Lamb's really, really good at. If you have one that's more recalcitrant, she can figure out a media on how that'll make it work. So with that expands the genetics that we can use for rootstock improvement. So I've got a strategy of crossing tetraploids again, because it's more powerful. You can select hybrids that have 3 or 4 rootstock genomes contributing to them. So we're screening both our diploids and tetraploids. Initially, we learned that the tetraploids also give us tree size control. I have a picture here of Valquarius on UFR3, which didn't turn out to be a good rootstock for a number of reasons, but you can see the kind of tree size and cropping you can get on these tetraploids. So they are really good for higher density plantings and smaller trees. But what I want to talk about is the mini Manhattan project that we've gone on, and we've known that time is the enemy, so we can't do anything fast enough. So think about historically how plant species survive. Out in the wild, you have genetic diversity. You have wild populations growing, and when there's a new disease that enters an area, it can wipe out the majority of all of the genotypes that exist there. But there's always 1 or 2 or a handful of genotypes that have adequate tolerance or adequate resistance. They intermate, form stronger babies, and then they grow up and they get that species through the bottleneck, and it can recover and maintain itself as a viable species in the environment. But because of man's destruction of all these areas that have the wild diversity, we've pretty much stopped this from happening. There's minimal wild diversity out there. There's still some out there, but it's less than what it always has been, and it's harder to find the things that you're looking for. But we feel like with the breeding programs and the tools available through biotechnology, we can sort of recreate this process and create thousands of hybrids that have different genetics that can be screened for the traits that we're interested in. So far, we screened about 20,000 seedlings through what I call the gauntlet. And when we first started this process, we were making one tree that we grafted and infected Valencia to and seeing what happened. And then we took the top of that tree that we selected and saved it to propagate the rootstock from. And we were putting one of each one out. And just by doing one of each one, this allowed us to have really, really high numbers. That was probably okay at the beginning because we were able to look at a whole lot of things. But now the program has kind of evolved, and I'll tell you some improvements that we've made. But we basically make all kinds of crosses, trying to make complementary combinations of the various traits. We screen them in calcareous Phytophthora inoculated soil. And if they can handle that, then they get selected and moved up to potting soil. And then they get grafted with the hot Valencia that's already infected with a high titer of sea loss bacteria. And then we monitor for the symptoms. And if they do well, they get moved out. And we were taking all these trees to Pico's farm at the USDA farm in Fort Pierce. So here's an example of our screening in the high pH calcareous soil inoculated with Phytophthora. And we've gotten better and better at selecting parents. And so when we first started doing this, we had crosses where only a handful of seedlings actually grew off. And now, we're getting crosses where almost all of them do. So here's how we do the initial screen. We graft a hot bud stick of the Valencia coming from an infected tree with a high titer determined by PCR. We use parafilm that we wrap Petri dishes with to wrap the bud stick. And this is really nice because you don't have to unwrap. The sprouts can push right through it because it's kind of flimsy stuff. But down at the bottom there where you see the graft just above the trifoliate leaves, that we use regular standard grafting tape there to hold the graft together. So a lot of times we'll see this, but you can't get too excited about that because a lot of times, even from the infected stick, the first flush that comes out will fool you and look pretty normal. But this is more like what you end up with. So these are gauntlet grafted seedlings from 3 different crosses, and it turned out that all 3 of these crosses are super interesting. We're getting super strong babies from all 3 of them. But you see on the right all of the duds, and you see all the left, all the ones that are good enough to go to Pico's farm. The rest of those get pitched. So all these go to Pico's farm. I mentioned these 3 crosses, and we have several hybrids from each one of these that are still looking fantastic, that are moving through the system and are actually going into some large-scale trials this spring. So they go to Pico's farm. They continue to segregate there. So you see a tree here in the middle. This is a former PhD student, Aditi Sapupte, who's working at Texas A&M now. But on the right there is a total dud. In the middle is a winner, and on the left is one that's trying to make up its mind. So this is again an infected Valencia growing off a normal tree on this complex rootstock that has myelin, pomelo, and 4 other things from Orange 14. And if the tree continues to do well, you see this, and this is what we're after. Starting out with a high infection. I want to mention this cross because it's Amlycarpa pomelo, a somatic hybrid, but it's crossed with a somatic hybrid of sour orange and rangpur. So sour orange and rangpur are 2 of the historically prominent rootstocks. Rangpur is still the #1 rootstock in Brazil, and it provides really good drought tolerance. So we had the somatic hybrid of sour rangpur in a very large rootstock trial with Harold McTeer with Roble Scion on Hatchnahall Road near the center. And at the time, the canker, we found 1 canker lesion in the middle of the trial. So they burnt, we couldn't stop. We tried our best to stop it, but they burnt the whole trial up. At that time, the sour rangpur somatic hybrid was the top performing rootstock in the trial for Roble Scion. And sour rangpur by itself did not pan out as a rootstock because it makes zygotic seed, so it was not amenable to seed propagation. But it also, the cuttings that we made from the sour rangpur were very slow growing in the nursery. It took a long time to make trees. So we used it as a parent because the seed trees at 3 locations were exceptionally tolerant. We still have 30-year-old seed trees that are still really nice looking trees that make nice seed fruit every year. So we've used them as a parent, and we've got these hybrids with Amlycarpa pomelo that a number of them are looking beautiful in the gauntlet. And this year, 3 of them made a lot of seeds. There was like 30 seeds per seed fruit, and we've grown out seedlings, and they're 100% uniform. So it's really exciting because we're going to capture this sour rangpur hybrid HLB tolerance in these hybrids, and we can propagate them by seed. So we've got 2 of these going out in a very large trial with a commercial grower this spring, and I'm very excited about it. Another thing you'll notice is that the fruit size on this collection of hybrids is always seems to be larger than what you see with other rootstocks. So again, I want to pound this idea into your head that the ultimate solution is the rootstock, because if you got one that can take care of the disease, you can grow whatever you want, including even honey murcots and all the grapefruit that you want in Hamlets, everything that you'd like to be growing. So I want to show you some PCR data. I know some people think it's worthwhile, and some people say you're wasting your time collecting PCR data. But I like it because it tells me what's going on in the top of the tree, and it tells me what's going on in the bottom of the tree. So I have a slide here. And of course, Mike Irie at the Southern Gardens Diagnostic Laboratory has been very helpful for doing tests on leaves. And then Lily Kano, the pathologist at the IREC, has helped us with roots. So the numbers you get for CT values are inversely proportional to the amount of bacteria, the bacterial titer in the tree. And so I have the categories of numbers up there. And I've actually adjusted these up in my head. So questionable infection now I would raise to 34, and then over 30, from 34 to 38 is probably a non-active infection. And then of course, 39 or 40 is probably means that there's no bacteria detected in the tree. So these, for me, these are useful numbers, and you'll see why in a minute. So here's the average numbers for all the commercial trees that were tested at these particular years at Southern Gardens with Mike Irie. And generally for a sweet orange tree, the CT values are in the mid-20s, which means there's a really high slug of bacteria in the trees. So that's a useful baseline to have for when you're looking for things that might alter this in the tree. So we've been screening the gauntlet trees. So the gauntlet trees, when you check them coming out of the greenhouse, they normally have a similar CT value to what you find in a commercial field tree in the mid-20s, 26, 28. But over time, we're seeing things in some of these gauntlet trees that are quite interesting. And a lot of times, the health of the tree is correlated with these numbers that we're seeing. So here you see out of about 800 rootstocks that made it to Pico's farm, we found quite a few that have no replication of the bacteria going on in the roots at all, which is interesting, because the first thing that was reported by Evan Johnson was swingled, as the bacteria goes down and replicates in the roots right away, and that probably helps contribute to the feeder root loss. So we've got a collection of these, and I can also tell you that Mongi and John and Fred Gamitter have some more interesting wide hybrids that we've made that are showing no replication in the roots, and it's giving us more candidates for wide hybrid rootstocks to test. But what I want to show you here is that some of these rootstocks also appear to be suppressing the replication of the bacteria in the scion. And the second one up there, S11 times 50-7612, is one that I'm particularly high on because this cross was really, really good. There's about a half a dozen hybrids in the gauntlet that are showing superior health, but this one, the tree is gorgeous at Pico's farm. I'll show you a picture of it. And it looks like it's making the sea loss bacteria in the scion go down to a level that may be below where it's going to cause any symptoms. It's a hybrid of a salt-tolerant Pomelobius Shaquasha mandarin crossed with trifoliate orange. So it's got really good rootstock pedigree on top of that. So it should make a very high quality orange in a smaller tree because the Shaquasha encourages precocious bearing. So here's another sibling of that in the field, 16.6. I also want to mention that I have both of these from this cross, the 16.6 and the 16.12 at Agra Melora, and they're doing very well in the micropropagation system. So these are going into a very large commercial trial with the commercial grower this spring. I thought I had a picture of that other one in the field, but I forgot to put it in. So I want to mention some other ways we're finding rootstocks. It's not just from the screening, but other rootstocks are emerging from various sources. So we're finding some mutants from field trials or from TC propagation. And there's also some hybrids from older trials that, you know, the cream rises to the top over time. So this is one that we're all very excited about. This is a mutant of the commercial rootstock X639 that we call STR41. And the trees you see here were planted between existing trees. So the test trees only are the ones that have the white wraps on them. You'll see one back there in the second row that's a runt. This is when they were planted again with HLB because they were leftovers from a nutrition experiment that we did in the greenhouse. We did infected Valencia on 5 rootstocks. And at the end of the nutrition program treatments in the greenhouse, we had trees that were too pretty to throw away. That's why we had to get a permit from the state, and we planted them out here as these resets. And so after 18 months, starting out with a CT value in the mid-20s again, I find this tree growing like a weed in the middle here that's got 50 fruits on it and flush coming out of all the shoots. How can that be from a tree that started with HLB? And it was supposed to be X639, and the other X639 trees weren't doing this. It was the only tree doing this. So we lopped it off, got it to sprout, and we recovered the tree here. You see John Chater holding it up. And so what happened next? Well, the sprouts came up. You see there on the right, and they had 40 CT values. There was no replication going on in the roots. So right after that, the grower we were working with sold the property for commercial development. So we had to go over there and dig up this single tree that existed in the world. We dug it up, and we had a hell of a time getting it out of the ground. So trees with HLB are not supposed to have a lot of feeder roots. Well, this thing had feeder roots out the wazoo, and we had to cut through 5 scaffold roots that were over an inch thick to get it out of the ground. We had a heck of a time. That was only 2 years old at the time. So we ran the rootstock on flow cytometer, and it kept coming up with a peak that was just slightly to the left of the diploid, which suggested that there may be some DNA missing, maybe a deletion of some sort. But we ran, in Fred's lab, we ran 13 SSR markers. They all matched X639. So we know it's a new cellar seedling of X639. But in the meantime, Fred has a federal grant working with some genomics people in Clemson University, and we had this thing sequenced. And Fred got the report back, and there's a 2-page long list of mutations in this particular new cellar seedling X639. So it's different, but it looks a lot like the traditional X639. So what have we done with it? So we had clean material. We got it going in Agromalora, and you'll see a picture on the right there. It's the most robust rootstock they have in their whole tissue culture micropropagation system. I've got an infected honey murcot planted on it that's doing remarkably well. If you can grow a honey murcot on a rootstock, you can grow anything because it's the most susceptible variety that we have available. And over here I have an EV that started out clean. It's taken off quite well. I've got a couple of these out. And I don't have pictures here, but I also inarched a compromised EV1 tree that was 2 years old, and the recovery of it so far has been quite remarkable. And hopefully I'm going to get a normal crop set on there, and I'll be sharing photos and fruit data on that this coming fall. So it's very amenable to propagation. We think it's subsequently going to be amenable to seed propagation because we did find one fruit on the seed tree that we transplanted, and it had about 12 seeds in it, but I picked it before it was ripe enough to get those to germinate because I was so anxious to see whether it had seeds or not. We have a cooperator named Jason Griffin, who is the owner of the property that the old Tropicana trial was on, 3 miles from the CREC at Lake Lowry Grove. And he went out on a limb and planted 1,200 OLL20 trees on this mutant without any IPCs. And I can tell you these trees are incredible so far. So we're all holding our breath, keeping our fingers crossed that this will continue to manifest itself in a positive way. But if you want to come see these trees, there's so many other good things to see at Jason's Grove that it's worth the trip, but we can show you. And they're putting on a heck of a spring flush right now, starting to pop out from these. And these went in as these little tiny Agromalora trees, and they're not even a whole year old yet because these were planted last June. So it's remarkable how they've done so far without IPCs. This is another one that's kind of emerged, very interesting because this is the tree from the St. Helena project. out in Dundee, and I've never seen symptomatic oranges on this rootstock. It's called MG11, and it has a really funny history too, because it's actually a seedling that my daughter made in her grade school science fair project about 20 years ago. But it's turning out that it makes the rootstock seed tree finally fruited, and it made seed, and the seeds produce uniform seedlings. And so it's going into a trial this spring as well. And it's a mandarin pomelo hybrid, so it should have very good tolerance to citrus blight as well. It also makes larger fruit, so we're testing it in cups to see if it can make some of the smaller mandarin fruit size larger, and the trees look gorgeous. We have the RES 1956 growing on this in the cups, along with some other rootstocks, and it looks like the best, most beautiful trees of any of the rootstocks we have in this little test so far. There's a whole bunch of fruit all on the tree from this season already. So I'm going to switch now and talk a little bit about Orange 14, which is the rootstock that we just released in time to get it included in the next round of craft. And it's from the same cross as UFR 1, 2, and 4. It's Nova Pomelo by Leo and Argentine trifoliate orange. So it's a complex tetraploid. It's amenable to seed propagation. We have seed source trees and we have seeds. And so it was made available for craft. So if you want to try it, seeds are available. And also I know one of the Southern Citrus Nursery also has seed trees producing seed, so it is available. So why did we release it? Because it's emerging as one of the more HLB tolerant selections. I didn't release it in the first batch because there was one 4-tree replication at St. Helena that always bothered me, but I think that's turning out to be a fluke of some sort. So anyway, here's a picture I took this fall of the seed tree. You can see how productive the seed tree is. And on the right, you can see the ton of seeds that it produces. Now you do have to rogue some zygotics out in the nursery. It's kind of like swingle, depending on the cross pollination. You can have 15–20% zygotics that need to be pulled out, but the majority of the seedlings are uniform and will make nice liners. This one emerged out at Ori Lee's in a blight test. This was before HLB, but when we were replacing trees that went out to blight, we were planting Valencian rough lemon adjacent to the test tree. And so here you can see that the Orange 14 was totally outperforming the 2 rough lemon trees before HLB. This is a photo taken in 2010, right when HLB was starting to make its way out to this grove east of St. Cloud, what we call the Alligator Grove. So here's a 7-year-old tree at Jason Griffin's, same place where I just showed you the mutant of the X639. These are 7-year-old trees in a 10-tree plot with OLL 10 sweet orange on them. They are loaded with very nice fruit. It's colored up now and probably will be harvested in the next few weeks. It's one of the strongest combinations there. And these trees are right next to trees that are 13 years old, and they've already reached the same size and have more fruit on them than most of the 13-year-old trees on various fruit stocks that are planted adjacent to them. Doing very, very well here. But one of the things that pushed us over the hump to release it was the performance of this rootstock. It got snuck into a craft project. It wasn't supposed to be available for a craft project, but it snuck its way in. And a really good local grower, James Shin, has this block of OLL 8s planted on it. These are 4-year-old trees in an area that has been hammered by Diaprepes phytophthora. So he says the Diaprepes, for some reason, don't like this block of trees. They're getting everything around it, but they don't like this block. And these trees are loaded, and they're probably going to be harvested in the next 2 weeks as well, as the crop's ready to go right now. I looked at them last week, and the fruit's still hanging on the trees. So it looks like it's a really good candidate for an area that has poorly drained soil or lowland soil that has an issue with the Diaprepes phytophthora complex. So what about now? So what's available commercially? This is some data from Ori Lee's trial. with OLLs, 2 years of yield data, but you can see that the orange 14, or UFR2, UFR5, white 1, which is a parent of a couple of the rootstocks I mentioned earlier. All these have done better than X639, Carrizo, and Swingle, without any psyllid control. This trial has not had psyllid control. Unfortunately, we lost this trial to development, so it doesn't exist anymore. John Chater has been working very closely with Jason Griffin, where we have the OOLs and Valencias on a number of rootstocks. But he did an economic analysis this past year, and he showed that OLL8 on UFR6 planted at optimum density can make you some money. And so you can take a look at the numbers here. The average yield was 1.37 boxes. The pound solids was just 5, and that's going to go up, especially if we figure out how to optimize the micronutrient nutrition. So you can end up making 23.98 a tree at 302 trees being the optimum density. You can pull in $7,200 per acre, and so what are your production costs? $2,000, take that off, you still make over $5,000 an acre. So there's a couple of other combinations in here that were profitable. So you can make money now if you plant the right combinations and you grow the trees with the right nutrition program. This is some data from a very large trial with Cutrale down at Venus. And this is only the first page. There's a zillion rootstocks in this trial. But again, I want to show you at the top of the list, white for yield, white 1, UFR 17, UFR 15, and orange 14, 3 things that are all available by seed. And of course, the white 1, I've got hybrids coming behind this. White 1 makes zygotic seedlings, so it hasn't been released because of that reason. We haven't tested to see how it does in tissue culture, but that's something worth doing as well. So again, if you go down the list, you'll see a lot of other rootstocks that are being planted that may not be as good yielders with HLB as these. So the conclusions from the rootstock screening work, we're finding a lot of hybrids that transmit some kind of tolerance to the scion. And be careful when somebody talks about a tolerant or resistant rootstock, because if it's not with a grafted tree, it doesn't mean anything. There's plenty of rootstocks that when they're ungrafted, they grow beautiful trees, but they don't necessarily transmit anything to a susceptible scion. We're breeding at the diploid and the tetraploid levels, and we're finding things from all this genetic diversity that we're looking for. I show the blue lobster because the blue lobster is a mutation that happens 1 out of, or 2 out of every 2 million lobsters. So with HLB, we don't know what the numbers are. Is it 1 out of 1,000, 1 out of 1,000,000 that we have to screen before we're going to find the answer? But the more you look at, the better the chances of finding that answer. So it's totally a numbers game. And again, the Manhattan project, the mini Manhattan project is well underway here. The ultimate answer. So what do you plant now? For scions, early oranges, you can try these 2 clones of Hamlin that are clearly better than the standard. For mid-season oranges, I like Balquarius, which is an early maturing clone of Valencia. It seems to be holding up better than Vernia, and it also has better fresh market potential. It's a more attractive orange for the fresh market. Late oranges, we got the new OLLs. We've got the new DC OLL clones, and there's more OLL clones coming along, including 2 that mature in January that you can watch out for. We have Valencia V965, which out yields the traditional Valencia. Coming soon, pay attention to this one. This is N40-16-711. It's the most HLB tolerant of 1,000 Valencia clones that we evaluated. It's cleaned up now. The original tree is on Swingle, is over 33 years old, still cranking out good fruit. We have resets on the parent tree program, budwood from this, that are 3 years old that look beautiful. Orange like hybrid, 1859 available now, 104 coming, and there's other ones behind that. Juice blending hybrids, I showed you two, C4 1042 and RBA 1318. For mandarins, Sugar Bell and Marathon work outside. RES 1956, great for cups, maybe outside. 1889, 39, the Gator Bikes, a home run for cups, maybe outside. And we got a couple other large fruited mandarin hybrids that are doing well in the cups as well. One of Fred's and two of mine that I didn't mention. For grapefruit, 914 is good. The C253, the early pink. But we're getting ready to release a Star Ruby quality dark red called N40 1611-7. It's a beautiful red blush grapefruit with the same color as Star Ruby, but the trees are much more greening tolerant, and they don't have that blotching problem that the Star Ruby's showing. We had 3 from the Mac trials from the Scott family, 4-year-old trees. We had fruit in our display. Grown outside, it was totally well received and really, really good. So that one's coming along for a traditional red. Lemons and limes, I showed you 4 new seedless selections and the C4 14 Fiestaville. And then, of course, there's Mongi's purple finger limes. Rootstocks. Of course, I'm biased to the CREC rootstocks. UFR6, very good for controlling the size of the tree and for cold hardiness, high soluble solids. 17 is emerging as a winner at multiple sites. UFR5 is totally outperforming US 942 at Eagle Lake for tree health, tree growth, and fruit quality with OLLs as a scion. UFR1s 1, 2, and 4 doing well, multiple locations, probably as good as the USDA rootstocks that are being widely planted ahead of them. And UFR 15 is one that people haven't paid attention to, but it's a one that should have very good blight tolerance. It produces a more vigorous tree. It's a Mandarin Pomelo hybrid with Pomelo and Clio. And it's the #1 yielding rootstock in the Millennium trial over at the IREC in Fort Pierce. And then you heard all about Orange 14 already. So we have choices. So for me, the immediate future is what I call the 3-legged stool. Get the good science together with the good rootstocks and pay attention to what's emerging as good nutrition information. Those 3 legs of the stool can work. I've got a picture here of a combination grown with good nutrition. Back in 2016, you see Allison standing next to some OLL8 on UFR4 trees. And then here in 2023, you see John Chater with Jim Graham, and you can go see these trees today, and they look just as good, and the fruit's orange right now and being ready to be harvested. So if you want to come see them now, you can get with us because they still look just as good as they did back in 2023. So there's sustained production. That's the ticket here. So thank you to a zillion people and sources of support to make all of this happen. So we're dedicated to get a lot of things that we have between third base and home delivered to y'all. And Fred and I are getting old, so we may end up finishing the job in wheelchairs, but we're still hard at it. Thanks for your attention.
Thank you, Dr. Grosser. Any question or comment? Dr. Grosser, is UF 950 still good?
US
UF 950 Mandarin.
Oh, 950 Mandarin. That's an interesting one. It's actually doing very well in North Florida. Down here in Central Florida, the trees look quite healthy, but you'll find some trees with a lot of symptomatic fruit. But in our trailer park, we found trees that had lovely fruit, and actually we put some of the trees fruit from outside in the last 2 years. We've had them in displays compared to cups fruit, and people picked the outside fruit over the cups fruit. But that was harvesting by avoiding the symptomatic fruit and harvesting off the trees that didn't have the symptoms. So I think it's very close to being a variety that can be grown successfully in Central Florida, but for sure you can grow really nice fruit up in the Quincy area.
Any question?
Subsequent questions, just holler at one of us and we'll be happy to tell you our best answer.
Again, if you are interested about CEUs, email me your name, email address, and license number. Thank you all for your participation and see you next time.